Chapter 2, Biopsychology. After studying Chapter 2, you should have a pretty good grasp on describing how neurons communicate, describing the process of action potential, describing the different neurotransmitters and their job in neurocommunication. Just recall the definition of biopsychology, that biopsychology is a branch of psychology that analyzes how the brain, neurotransmitters, and other aspects of our biology influence our behaviors, thoughts, and feelings. This week we are focusing on neurons and neurotransmitters, the brain structures and other areas in our body that interact with the brain structures will be explored in upcoming weeks. Before we get too deep in our study of more complex things like action potential and neurotransmitters and the different kinds of neurons and their jobs in our central nervous system, I want to take a minute and just reflect upon the fact that these cells are really quite extraordinary. The quote from your textbook on page 24 says that neurons are deceptively simple in action but impressively complex in function. Neurons are specialized cells. They carry messages back and forth between the brain, the spinal cord, and throughout the body. They collect sensory information from the environment via the sensory neurons. Neurons result in your thoughts, your feelings, or your actions. They transmit messages throughout the body, muscles, organs, and skeletal system. They communicate internally within the brain and the spinal cord. And they communicate with each other to control bodily functions at a muscular, skeletal, and glandular level. Neurons are definitely extraordinary little cells. They're not simple message conductors, but information integrators. Neurons use information to make decisions that determine whether to fire or not. Think about it. A simple trigger of the soma sends a signal. That signal begins a chain of signals that spreads across multiple neurons that will eventually result in a behavior or a thought or elicit a memory or allow you to solve a problem. It makes us who we are. We wouldn't be the dominant species if it wasn't for the communication of these little cells. The human body is made up of billions of neurons. And for this class, we're primarily going to be focusing on motor, inter, and sensory neurons. But there are far more neurons that make us who we are and able to do the things that we do. In this picture, you see what is a typical multipolar neuron. It's your typical motor neuron. And I just want to kind of walk you through the different parts of this neuron. It starts at the presynaptic cell. You can notice the dendrites coming off of the cell body. Inside the cell body is the nucleus. Coming off of the cell body and attaching to the axon is the axon hillock. Your axon travels quite a distance. It's covered in myelin, and the gaps in the myelin are called the nodes of Ranvier. It also shows you how the signal is traveling down the axon to the synaptic terminals or vesicles. The synapse is the space in between the two neurons. They are not touching. And then the postsynaptic cell is on the end of the neuron, ready to receive the message from the presynaptic cell. So motor neurons are responsible for allowing and providing us with the ability to move. They receive instructions from the brain and the spinal cord and carry these messages to your muscular units and your skeletal systems. Motor neurons are long. They can go the entire length of your leg, and they carry messages all throughout your body. So another type of neuron is the sensory neuron. The sensory neurons can be either a bipolar neuron or a unipolar neuron. The difference between them is that these neurons have the axon that extends in both directions from the soma, and in a unipolar neuron, the soma is actually detached from the axon and it sits up on like a little stalk. Sensory neurons take incoming stimuli from the environment via your senses, and they send signals to the interneurons. These interneurons that are located in your brain and your spinal cord, they communicate and they allow for responses to happen to these external stimuli. For example, they might trigger a motor neuron to move your arm to scratch and itch, or to move your arm to shade your eyes from bright sunlight. It all starts with the sensory neuron. The sensory neuron receives that information, sends it to the interneurons where they do the final processing and make the final decisions with how you should react to your environment. Interneurons are multipolar neurons because there are many extensions that come out from the soma. However, interneurons have a short or even no axon. Their job is to communicate with neurons that are close by them, so their signals do not travel great distances. Interneurons reside in the brain and the spinal column, and interneurons communicate within your central nervous system to relay messages from sensory neurons to other parts of your body so that you can react accordingly. They are what I consider the bridge between your motor and your sensory neurons. Now there's basic neural communication, and it begins at the dendrites. The role of the dendrites is to collect the information from other neurons and to pass this information on to the soma. The soma, or the cell body, will then evaluate the strength of the signal, if it's excitatory or inhibitory. Does this signal reach the threshold for activation, or is it not strong enough? If it does reach the threshold for activation, then the axon opens and allows the electrical charge to begin. This is called action potential. Now, when this excitatory stimulation causes action potential, it starts as a depolarization of the neuron. Once that starts, it will cause a shift in charge within the soma, and this excitement exceeds the threshold, resulting in action potential. The sodium channels will open, and in an explosion-like fashion, the ions will enter into the axon. This opening of the sodium channels travels the length of the axon, gaining force and speed by the insulator myelin. This buildup allows for the action potential to jump from node of Ranvier to node of Ranvier to node of Ranvier, where there are more sodium channels in that area. Finally, this signal will reach the synaptic terminal or the vesicles. It is important to remember that this entire process is an all or nothing process. In healthy functioning neurons, once action potential begins, it finishes. It's non-decremental, it's ungraded, the neuron will fire until the message has been sent. I said healthy neurons because there are some cases where there are structural abnormalities, diseases, disorders that can affect the flow of action potential. For example, multiple sclerosis is just one example. MS is known for the depletion of myelin all along the axons of nerve cells, and without this myelin, the nerves cannot send their signal effectively, causing transmission issues and just overall problems with cellular communication. On this slide, there is a link to a YouTube video. It's about 4 minutes long that will give some more information on neurocommunication. Please just copy and paste this address into your web browser and hit enter, and you should be able to access this video for further explanation. So when your cells aren't communicating, your neurons are just at what they call resting potential. Neurons are charged particles. The charged layers of the membranes, they hold the cell together by either attraction or repulsion to the seawater-like fluid inside the cell. These opposite charges will cause the cells to come together, or the ions to come together, creating a wall or a membrane that controls what is allowed in and out of the cell. Polarization happens when there's a difference in charge inside and outside of the cell. For example, the outside of the cell is typically positively charged, and the inside of the cell is typically more of a negative charged area. When the charges change, they create a certain level of excitement, and then the cell shifts from resting potential into an action potential. Now, resting potential is generally at a negative 70 millivolts. It's important to know that this does not mean that it's negatively charged. This is just the comparison or the difference between the inside of the membrane and the outside of the membrane. There's unequal ion distribution, but the ion itself is not negatively charged. Action potential happens when the neuron gets excited and partial depolarization occurs. If partial or local stimulation exceeds the threshold, then the sodium channels will open up. And that's when there's an abrupt depolarization of the neuron. The sodium ions will flood into the axon to a volume of 500 times greater than what it was at resting potential. And now the inside of the cell is positive. This causes a chain reaction along the axon, opening up the sodium channels all along the nodes of Ranvier, allowing the electrical charge to flow. The signal is insulated and gains more speed due to the myelin. The absorption of the energy from the myelin allows the signal to travel, like I said, jumping from the node of Ranvier to node of Ranvier. This is how the signal gets revitalized, and this is what keeps it going and keeps it strong so that it can travel the length of the axon. Remember, some axons could be the length of your leg. The refractory period is what comes after. It's a time when the signal has been sent and the cell cannot fire again unless it receives a stronger than threshold stimuli. This refractory period is very important because it means that cells cannot be overstimulated and firing. Overfiring and overstimulation of cells could cause, mayhem inside of our central nervous system. And just in case I talked too fast or missed anything, this video will also help explain the change from resting potential to action potential. Again, just copy and paste this into your web browser. There are more glial cells in our central nervous system than there are neurons. And this is because glial cells have many important functions in maintaining healthy neurons and in assisting with neuron communication. Glial cells provide energy to the neuron. They support the neurons by being cleaner cells that go in and help clean up the synapses when there's extra neurotransmitters hanging around. They also support neurons because they guide them during development, and they bring them to their correct location in the central nervous system. Glial cells also help make myelin, which is very important, as we know, for allowing neurons to effectively communicate by acting as an insulator. And all of these jobs that these glial cells do help strengthen neural connections. It's been shown that when glial cells are removed from the body or destroyed or depleted, that synapses are actually lost. Okay, so now the action potential has traveled the length of the axon, and it has reached the synaptic vesicles and has sent neurotransmitters across the synaptic gap to the postsynaptic neuron. There are vesicles on the postsynaptic neuron that are specialized for each neurotransmitter. They have special shapes and locations that fit with the neurotransmitter that's being fired. When the action potential arrives at these vesicles, they open in a process called exocytosis. There are special chemical receptors on the postsynaptic neuron, as I just mentioned, and the bound neurotransmitter continues the signal that was sent from the presynaptic neuron, again then causing the dendrites to send the signal into the soma to activate the axon hillock and to then continue the signal through yet another neuron. This eventually will result in a reaction. Keep in mind that these are graded potentials. They can be excitatory or inhibitory. Excitatory postsynaptic potentials are called ESPs, and inhibitory postsynaptic potentials are called ISPs. Keep this in mind, though, that just because something is called excitatory, it doesn't always mean activity. And just because something is inhibitory doesn't always mean that it's inactivity. For example, Ritalin is the medicine used to treat ADHD. It's a stimulant, and it actually increases nervous system activity, but it has a calming effect because it speeds up the activity in the frontal lobe, allowing the person who suffers from the ADHD to have more activity in their frontal lobe, allowing them to make better decisions and be able to reason better and to be able to problem solve better and so on and so forth. So it is a calming type of neurotransmitter. So it is called an ISP, even though it's a form of stimulation. So one neuron can get input from thousands of other neurons and can get input from up to 10,000 different connections. Think about what are the benefits of having multiple connections to one neuron. The first benefit is postsynaptic integration, and this occurs at the axon hillock. What this is the combination of action potentials from many different neurons. This limits accidental firing and responses from spontaneous activity, meaning that if one neuron accidentally fired, the postsynaptic neuron wouldn't release the signal that it was sent. It would need more than one neuron to send it the signal in order for it to then send the signal on to other neurons. Another benefit to these connections is spatial summation. This combines potentials from other locations that come in at the same time. And then there's temporal summation, which combines potentials that come at staggered intervals. So even neurons cannot stay active. Even neurons need to rest. They rest during a process of reuptake. And during reuptake, excess neurotransmitters that are floating around in the synapse are taken back into the vesicles of the presynaptic neuron for later use. Also floating around to help the process so that the neurons can get their rest are the glial cells. As we mentioned before, they're around to clean up the excess neurotransmitters, and they're also hanging around the synapse to stop neurotransmitters from escaping and potentially getting in the way of other communications with other neural networks. There are other neurons that have the job of just regulating the presynaptic neural release by regulating calcium from the presynaptic neuron, and there are autoreceptor neurons that monitor neurotransmitters in the synapses. The job of all of these neurons and cells is to essentially help clean up and end the transmission or the communication so that the neurons can go through their restful stage. So there are many neurotransmitters that are fired within our central nervous system and within our neural networks. On this slide, you'll just see some of the basic neurotransmitters that we'll be talking about throughout this course. The first one is acetylcholine. We have serotonin, dopamine, norepinephrine, epinephrine, glutamate, GABA, glycine, and then there are endorphins, substance P, and neuropeptide Y. So I want you to notice that not all of these are neurotransmitters. There are some neuropeptides, neuropeptide Y. Neuropeptides are chemicals that act as neurotransmitters. And just to break it down even more, peptides are chains of amino acids. Proteins are longer chains of amino acids. So they're all related to proteins. Neuropeptides can be released directly into the blood to activate receptors. They can also be released into the brain, where they will directly activate brain receptors. Below there is a link for additional information on neurotransmitters, amino acids, and neuropeptides for you to look through at your leisure.